we continue with our lecture. So in the exponential phase or the lag phase, the bacteria multiplies at a constant rate. And that rate at which the bacteria multiplies is not limited by any environmental factor except by its own genetic potential. So you need to understand that during this phase, growth rate is constant. So the bacteria is not limited by any external factor. And you need to understand that all the cells in this phase are alive, and there are no spores that form in this phase. And again, you need to understand that it is in this phase that the bacteria is highly susceptible to antibiotics, because almost any chemical that you can bring, it can affect the bacteria, because the bacteria is actively carrying out metabolism. If you talk about penicillins, for example, disinfectants, they are very effective in this stage of growth. Then this third phase is what we call the stationary phase. In this phase, there is nutrient depletion and accumulation of toxic waste products that have started. So you need to understand, because of the accumulation of toxic waste products and also the depletion of nutrients, the bacteria begins to slow down in terms of multiplication. Those that form spores begin to form spores, and there is no net increase in cell number. In short, there is no growth that is happening. And this is because the rate of growth or the rate of increase of bacterial cells equals the rate at which the bacteria dies. So because of that, you will have this phase as a stationary, meaning there's no change, there's no net change that is happening in terms of the number of bacterial cells. Then the death phase, the last one. In this phase, there is total depletion of nutrients, and the cells can no longer maintain viability, and the numbers begin to decrease in proportion. So they begin to die at a constant rate, continually, because of the increase in waste products and also the depletion of nutrients. Just in order for us to appreciate this, maybe let's look at the curve. This is the lag phase, and this is the exponential phase, and this is the stationary. So as you can see from the structure, all these phases are showing you something. The shape of this phase, the shape of this curve is telling you something. Remember, this is the plot of time, the number of bacterial cells against time. So if you look on the y-axis, we have the number of cells of bacteria. Then on the x-axis, we have time. So as you can see in the lag phase, time is increasing, but the cells are not increasing, meaning there is no growth that is happening. This growth is an increase in the number of bacterial cells. But when you look at the exponential, it is straight, meaning there is rapid increase, gradual increase in number of bacterial cells, inhibited only by its own potential. So during the log phase, this is the phase in which you can study the properties of the bacteria, and it's a phase in which you can easily kill the bacteria. So generation time can be determined in the log phase. You cannot determine generation time in stationary or lag phase or decline phase. You cannot determine generation time during those phases. So appreciate this and relate it to what I just explained on the previous slide. Then among the factors that affect the growth of bacteria, one of them is temperature, oxygen requirement, pH, availability of water, and also the nutrients. So with respect to temperature, there are bacteria that require cold temperatures. Some of them, they just require warm temperatures, others hot temperatures. So those that infect us, usually they are found in this range between cold and hot, just in the middle, because this is where our body temperature falls. So bacteria whose growth is best between 25 and 40 degrees are known as the mesophiles. Then those that require cold temperatures, they are known as cycrophiles or cycrotrophs. So you need to understand that these bacteria are a problem when it comes to spoiling the food we keep in our fridge, because for them their growth temperature is below our body temperature, and they are able to grow at the temperature at which we keep our food in the fridge. So they are the culprits when it comes to causing spoilage of food that you have kept so that you can eat tomorrow. These bacteria are a problem. Then you need to understand that most of these bacteria, they don't cause infections, except for one bacteria that is able to grow as a mesophile and also as a cycrotroph. This bacteria is Listeria monocytogenes, the causative agent of listeriosis. Then the other thing you need to appreciate is classification of bacteria according to the oxygen requirements and also pH. So please, in your own time, just try to appreciate this.